Testing, testing. Oh, good. Okay, hello. Hello. Welcome to our new room. I hope you don't like it too much because we're only going to be in here this one time. We're kind of like a wandering tribe this year. It's been very hard to find good lecture halls, but we're here now, and I want to introduce, first of all, the topic of this year's lectures, because this is the first lecture of the year, and then introduce our speaker. Okay, so the topic is the neuro in neuropsychiatry. Like, is the neuro actually doing any work for neuropsychiatry, or are psychiatrists just feeling like, oh, they need an image overhaul, neuroscience is cool. Let's refer to ourselves as neuropsychiatrists. Okay, that's a little cynical, and absolutely not fair. On the other hand, if you look at the state of neuropsychiatry, there's not a lot of neuro to be found in it. The DSM, the Diagnostic Statistical Manual of the American Psychiatric Association, which defines all of the different disorders of psychiatry, is based totally on behavior. The diagnostic criteria for any psychiatric syndrome, not blood tests, not brain scans, behavior. So that's not too neuro. The treatments for psychiatric disorders are mostly drugs, also some talking therapy, some very cutting-edge stuff, which we'll get to in a minute, with brain stimulation. But these drugs, okay, you might say, well, drugs, that's a brain thing, right? I mean, these drugs are working on the brain, not the kidneys. But, in fact, many of these drugs were discovered essentially by accident. The meds had side effects relative to what they were being developed for. Stimulants, which are now used widely to treat ADHD, for example, they were developed as an antihistamine. And back in the days when scientists tested things on themselves, which, be warned, the IRB does not look kindly on that. You can't do that either. But he took some amphetamine, and he was like, I feel very wide awake and alert, and this is really great. I don't know if he had a stuffed nose or not, but he definitely found increased attention and focus and motivation. The earliest set of antidepressant medications were discovered because one of that class was used to treat patients with tuberculosis, and then they noticed that all these TB patients in the sanitarium were like suddenly really cheerful and singing songs in the hallways and stuff. And some courageous doctor decided to do a clinical trial for depression and found that it worked. Okay, so this is hardly neuroscientists poring over their data and saying, well, according to my theory, a molecule like this should improve concentration or improve mood. In fact, with SSRIs, people still don't really know how they work. That's like an ongoing topic of research. It's not, like the drug companies say, that you don't have enough serotonin in your synapses. Okay, I am digressing, which means I should probably stick with this thing. So this is the kind of question that we like to ask and consider with our fellow Penn community. Namely, is neuroscience, is it really adding anything to the many fields of society in which it is now being used? Or is it just, neuroscience is cool, we're going to get on the bandwagon? This year, we are going to hear from people who are pioneering new neuroscience developments that will help alleviate suffering in psychiatric patients. This is really kind of a sea change in the way the way psychiatric drugs are introduced, or psychiatric treatments in general, I should say. This work, I think, and I'll wait to hear what you say, because you're the expert, but I think this work would still be called translational. Okay, it's, people cannot call Dr. Halpern's office up and say, my doctor says I have an impulse control disorder, so I'd like to make an appointment to have you operate, right? On the other hand, this is pretty advanced translational research, translation from basic science to the real-world application, in the sense that there are now clinical trials with humans being conducted. So they're getting close, right? So this is very encouraging and exciting. We're going to hear from several people whose work conforms to that description, as well as a couple of skeptical voices for balance. But today we're going to hear from, I'm sorry if this sounds like I'm just sucking up, but as far as I can tell, and I'm not an expert, but this is the most promising, truly novel use of neuroscience to treat a psychiatric symptom. So I'll stop there with the grand theme and just tell you a little bit about our speaker, Casey Halpern. He is a Penn guy from all along. He did his undergraduate work here. He did his medical degree here. He did his residency here. Then those rats at Stanford recruited him away to a faculty position. Happy days. We got him back at Penn as of a couple of years ago. He is the chief of stereotactic and functional neurosurgery, and he has been innovating novel treatments for psychiatric disorders, particularly loss of controlled eating. And I'm going to just end my intro there and give you Casey.
It's such a pleasure to be here. I guess you can hear me okay. I was telling Dr. Perelman that I used to study in this room when I was in college, and my wife was in Wharton, so I could sneak into Steiny D. And they had coffee and things like that until like 2 a.m. So this was always the place to study. Downstairs we would socialize, and I always found that distracting. I'd rather not be in the basement of Steiny D and out having fun, but this was the room where we could study and have peace and quiet. So I don't know if undergrads are here at all, but that was a fun thing to do when I was studying for my exams. It's great to be back. My practice is a Parkinson's-focused clinical practice. I saw deep brain stimulation for Parkinson's disease over 20 years ago for the first time when I was in college at Pennsylvania Hospital, where I came from today. So I'm sorry I'm a little bit late. I was doing a deep brain stimulation surgery. I perform that surgery now in the room that I first saw it in on the 8th floor of the hospital, performed at that time by my mentor, Gordon Baltuck. So it's a real privilege to be back from Stanford, as sunny and great weather it is that they have there, to practice the same procedure that I learned here in the very same room. HUP, or the pavilion that you have here on campus, is where we practice a lot of neurosurgery, but the Parkinson's Center is at Pennsylvania Hospital, always has been, and at the end of my talk I'll sort of circle back to that history and a very special hospital, which is Pennsylvania Hospital. If you haven't been there, it has a museum, it's America's first hospital, and it has very relevant history to today's topic. So we agreed on a title together. I speak on brain stimulation for psychiatric disorders a lot, and non-invasive, invasive, sort of combinations of the two. And obviously, the goal is to develop a brain stimulation strategy that can be scaled for the large masses of patients that suffer from these problems. So when you hear me talk about it, know, without me necessarily saying it every time, this is for the patients who have failed everything. And if you think about that patient, and many of you probably know a patient like this. This could be a patient with severe eating disorder, severe OCD, obesity, Tourette's syndrome. Think of the most severe case and a patient who has tried everything. And this is a young adult, and they have a long life to live because otherwise they're very healthy. And what are we going to do? And so that's where a procedure like deep brain stimulation could be something that we think could be helpful. When I was in college, I wanted to start doing this for patients with cocaine addiction and obesity. But my mentors at the time, Tracy Bale, John O'Riordan, people that, some people are still here, some have moved on, some have retired, advised I start in the mouse. And they said, Casey, you're just in college. You're not going to be able to start a clinical trial. So why don't you find a mentor in the mouse world? And this was Tracy Bale over in the animal biology department. And see if you can actually study what your hypothesis is in a mouse before you do it in a human. I thought that was a good idea. So that was back in like 2008, 2009. So I've been working on this for a long time. Penn, as always, has us report our disclosures. None of them are relevant to today's talk. I do have research support for everything I'm going to speak on today from the NIH and the Foundation for OCD Research. So everyone knows that obesity is an extremely common problem. What is particularly important to note is that the epidemic of obesity is relatively recent. In 1990, it was half of what it is now in this country. And it is really important to recognize that underrepresented minorities are impacted by this problem more so than non-underrepresented minorities. I realized this over 10 years ago, and I put a team of thought partners together when I was just leaving college, moving into medical school, and just created this idea that deep brain stimulation could be helpful for certain patients with obesity. And these are probably patients who have tried everything, including gastric bypass surgery. I was really interested in studying a feature of obesity that is not necessarily common to all patients with obesity. Remember, obesity is something that you can see. It's the result of a problem or many problems, probably many. But I was interested in loss of control or behavioral disinhibition or impulsivity or compulsion, and finding a part of the brain that was involved in those problems, maybe a habit even. We can talk about that if you'd like. And actually, Alan Rosenquist, who was a dean in the medical school at the time, one of the deans, there's many, he told me, he was a neuroanatomist, he said, Casey, I think you should study the nucleus accumbens, because if you target the appetite center of the brain, which a lot of people study in the context of obesity, including a lot of people at Penn, you might forget about all these other disorders like addiction and OCD that suffer from similar types of compulsion. So go after the nucleus accumbens. And it was really just that. And so then I found Tracy Bale's lab, and we started investigating this common molecular pathway of reward, because all of these rewards or stimulants or stimuli have different mechanisms in how they create that rewarding or reinforcing property, that sort of sense of joy that you can feel when you consume the reward. Let's say it's a cheesesteak. And we can argue about which cheesesteak. I love Angelo's cheesesteaks on 9th and Christian, by far better than anything in deeper South Philadelphia. But all of these rewards modulate the reward circuits of the brain and converge on the dopaminergic structures, the neurons that project from the midbrain to the nucleus accumbens. And given you have this sort of pervasive problem, not just societally, but in the brain, and it impacts the brain in so many different ways, however, it all converges here. To me, this is a high-impact opportunity to help society in a major way if we can have a scientific breakthrough. And so this is kind of how I started, and this is what I've been working on ever since. In 2013, this was about 2 years' worth of work, but during my postdoctoral research period with Tracy Bale, I placed a deep brain stimulation electrode in the mouse nucleus accumbens. I was a neurosurgery resident at the time doing brain surgery in humans. I had to learn how to do brain surgery in mice. It was not as technically challenging as I was worried. We studied them in their home cage because they're very stressed animals, so we wanted them to feel comfortable. And you can see at the bottom here, right here, these are just two examples of when we stimulated, this is the averaged effect, when we stimulated the nucleus accumbens, there was a suppression in the amount of high-fat food that the mouse was eating. And we could reproduce that every day. We could do it for months at a time, and I did it for months at a time, which is how we got it published in the Journal of Neuroscience. And we studied its mechanisms pharmacologically a bit as well. The problem at this point was that deep brain stimulation as a therapy was very different to how I did it in mice. Deep brain stimulation as a therapy is typically like a pacemaker for the brain, where it just stimulates all the time. But in the mice, I was stimulating when the high-fat food was present, only when they were about to binge. So what we wanted to build was this concept of a smart stimulator, something that would stimulate automatically, and not stimulate most of the time, because people that have binge eating disorder, a lot of the time actually don't have a problem. It's only in those moments of vulnerability that they suffer. So we wanted to prevent those moments, but not necessarily interrupt normal reward sensations. So when I got to Stanford as faculty, I was able to get a K award with Rob Malenka as a mentor. I don't know if you know the name. Rob is sort of the living father of addiction neuroscience. Probably may not win the Nobel Prize, but I would argue he probably should. Optogenetics, which is a really important technique in neuroscience these days, was discovered in his lab by his postdoc, Carl Daseroth. But Rob Malenka never takes credit for that discovery. Carl Daseroth will probably win the Nobel Prize for that discovery one day soon. In any case, I went to Stanford partly to work with Rob because he's a wonderful mentor. And we agreed that one way to automate this would be to see if there is an electrical signal in the nucleus accumbens that an electrode could capture, and then stimulate when that signal was present. And there was already evidence from fMRI and other types of neurochemical assays in the nucleus accumbens in awake-behaving mice that there would be something there. So it was a pretty simple hypothesis and a fairly straightforward experiment to carry out. And indeed, every time we exposed mice to high-fat food, over time, they would develop this increased power in the low-frequency range. This is a LFP, or low field potential recording. And the power would start about 1.5 seconds before time zero, which is the moment of the bite. In fact, I started at that moment a research program at Stanford called BITES, because we believe it's all about the moment of the bite. And as you can see, when regular food is given to the mice, there's really no signal here that a device could use to trigger stimulation. So it really wasn't expected that regular food would induce any therapy, only exposure to high-fat food. And this was our model of binge eating behavior. It's a mouse model. It's not a human condition, but it's as close as you can get. And indeed, compared to other rewarding assays, the high-fat food would strikingly increase the power in this low-frequency range. And so the simple hypothesis was that we could use a device to detect this signal in red, and then in red down here, trigger stimulation. And we compared it to standard deep brain stimulation like we did originally here at Penn when I was a postdoc. And it was better, admittedly. It was very similar to stimulating when the mouse was approaching the food. So my postdoc would actually sit in a nearby room under video surveillance watching the mouse, and as the mouse would approach the food, he would deliver stimulation sort of automatically during the approach to that high-fat food. And that manual stimulation in orange worked the best. But we couldn't convince the NIH that we would let people self-stimulate, although I am still kind of curious about that strategy. We can address that in another talk. So RNS, or responsive neurostimulation, I now call it responsive deep brain stimulation for a variety of reasons, seems to be an important translational opportunity. And this is one of the first mice. Typically, the mouse would eat this high-fat food out of our hands, because it was like sweet butter. But here you'll see the food gets delivered here, and in black, the device is off, but all of a sudden it's on, because this low-frequency signal over here on the bottom left was detected, and it would trigger stimulation. You can see the mouse doesn't really seem perturbed, or there's no really adverse effect here. But as the device shuts off, the mouse then approaches again and it gets turned on again. And in this case, it's going to get turned on a little bit longer because the detection kept coming. And the mouse is walking around kind of freely here without any obvious side effect. Obviously, the mouse can't tell us what it's feeling. So leveraging the Malenkov lab, after we reproduced this in many mice, I proposed to do this work in my K Award. What I didn't realize was how sophisticated these postdocs were in Rob Malenkov's lab. Rob Malenkov has a dynasty lab of 20 postdocs, all of whom are independent scientists that could out-science me any day of the week. And so we collaborated. So Dan Christoffel, who's now faculty at UNC, did a lot of this work with me, and I got to be sort of a co-mentor in a way. But I couldn't have done this without them. And what Dan demonstrated was, and he went after every circuits I didn't even heard of. He was actually using optogenetics to try to drive and inhibit these circuits. Because obviously electrical stimulation may be stimulating circuits, but it could also be inhibiting because electrical stimulation, who knows what it's doing in the human brain or in the mouse brain. But the pathway that he was most interested in was this inhibitory control pathway from the prefrontal cortex down to the nucleus accumbens, specifically the ventral medial prefrontal cortex and the nucleus accumbens shell subregion. Same region that I targeted with deep brain stimulation. And this was a sophisticated design where he would actually inject the virus into the prefrontal cortex and then would actually activate the channel rhodopsin and depolarize cells. Is this something you guys are familiar with, optogenetics? Okay, great. Some said yes, some said no. Suffice it to say, this is a technique to use light to either inhibit or activate neurons. And so that mechanistic robustness is why Carl Daseroff is not so famous. He's made a lot of other discoveries, by the way, but that's the one he's most famous for. So we would actually activate those terminals of the prefrontal cortex in the nucleus accumbens itself. And by doing that, we would, down here in red, mimic the effect of deep brain stimulation. We were able to actually have the opposite effect, which is really amazing. If you think about these mice binging, or if you think about yourself after a binge, when you eat a lot of food, like when I feel hungry between surgeries, I eat a lot of food. But when I'm done eating, there's no way I can eat more. But these mice, we could actually drive them to eat more beyond capacity with other techniques. But this was the technique that seemed to mimic what deep brain stimulation was doing. So now we have a hypothesis here that if we can drive this circuit, targeting this region of the nucleus accumbens, perhaps we can actually help patients with loss of control eating, a very common feature of obesity and eating disorders. In mice, we call that hedonic feeding, by the way. We don't really use that term in humans, but hedonic feeding sort of means pleasure eating. It's sort of oversimplified. We tend to call it loss of control eating, which is a construct that's very common in the eating disorder literature. So what we didn't know was whether this circuit, the prefrontal accumbens circuit, was abnormal in humans that have obesity or humans that have eating disorders. So that was something that my lab had to tackle now that we at least had a hypothesis from a mouse study that we had not only a potential translational finding, but a mechanistic understanding of it to a certain extent. So using tractography, which my lab has sort of now adopted as one of our main methods of sort of scientific circuit disruption and understanding concepts in ways that we've never really understood before, we were able to actually parcel out two regions of the human nucleus accumbens in red and yellow. And why was that critical? Well, because in a mouse, using histology, because you can kill a mouse after a surgery and actually see where your electrode is. In a human, obviously, we don't do that. We use MRI scanning to do that. And we wanted to have some technique to confirm that our electrodes were in the homologous structure within the human brain. And actually, if you know the name Suzanne Haber, Suzanne told me, she's a very famous neuroanatomist. She actually said to me that this brain region of the mouse is probably, and she said probably, the only mouse brain region that shares homology with the human brain. So that made me a little bit lucky. Alan Rosenquist didn't tell me that, but he probably knew that when he advised that I use this structure to study, because it made my mouse work so much more translational. And here you can see two structures within the human nucleus accumbens, just like we see in mice. And they actually are very similar to the subregions that you see in a primate using histology. I'll mention that, like in a mouse, this yellow subregion exhibits stronger connectivity to the ventral medial prefrontal cortex, the same structure that was studied in a mouse, than the more dorsal structure in red. And that is very similar and homologous to what we see in mice. So now we, luckily through our collaborations, this paper is now actually accepted in molecular psychiatry. This is an eating disorder expert, Kara Bowen at Stanford. Jennifer McNabb is one of the most brilliant MRI physicists I've ever worked with, also at Stanford. We were able to acquire a data set of patients with obesity and binge eating disorder, a very common comorbidity. And we found that the connectivity between the nucleus accumbens and the ventral medial prefrontal cortex went down as BMI went up. So there was this clear, what we can say, a perturbation of this circuit. We don't know if it's related or due to the obesity, but certainly this is a finding that is in line with the hypothesis that we were deriving from mice. I maintain my collaborations with Jim G. and Murray Grossman. These are wonderful collaborators. Murray, we have a long history together, recently passed away. Murray, when he passed away, was the most federally funded neurologist at Penn. Now he has transitioned all of his grants to his trainees and collaborators at Penn. But he was my mentor and also my soccer coach growing up. And we have a long, fun stories to share, but I don't think we have time tonight. He's also a VBM expert, voxel-based morphometry expert, and they actually offered to help us analyze these brains and look at the gray matter of the prefrontal cortex to see if it exhibited any differences based on BMI. And indeed, there was a thinning of the prefrontal cortex as BMI increased. And then, not surprisingly, a strong, significant correlation between the thickness of the gray matter and this connectivity, suggesting a relationship between the connectivity and the intactness of the prefrontal cortex of these patients. This is resting state functional connectivity, just demonstrating that this is not just a structural difference, but also a functional difference between the two. So this sort of perturbed circuit that we now can potentially rescue with a intervention. We did this acutely with neurostimulation. So with electrodes sitting in the shell of the nucleus accumbens, and we only activated these electrodes here in red, we were able to induce relatively rapid decreases in cravings, improvements in self-reported self-control, and even over the course of a few months, less body weight in a very severe obese patient. That was one of our first human patients that we studied. And this was published by Daniel Barbosa, who sadly left Penn recently for a residency position at MUSC. So while I was at Stanford, this is one of my last mouse papers. I now do entirely human work. One of my last mouse papers we published in PNS, where we demonstrated that we wanted to know what happens to the neurochemical environment of the nucleus accumbens when we stimulate it. And when we stimulate it continuously in red, you can see in red, there's this, it's actually at time 0, there's a spike in red. And that red represents calcium activity. This is fibrophotometry, measuring calcium signal as a proxy for neural activity. Also invented by Carl Taysroth at Stanford. This is sort of the opposite of optogenetics. In any case, what he found was that, and what my team found, was that when we stimulated this area with deep brain stimulation, the calcium signal went up briefly, but then it kind of got suppressed and never went up again. So it suggests that when you do continuous stimulation to this area, it has sort of a functional lesion approach. It quiets it, because you expect there to be some activity. But if you do this sort of automated stimulation strategy, so stimulate, if you remember when that frequency was really low and strongly powerful in red, when the device stimulates in response to that automated signal, when mice were approaching the high-fat food, there's this intermittent increase in calcium spiking every time the stimulation gets delivered. So it actually speaks to the fact that we can actually deliver electrical stimulation and actually stimulate, at least within the locus of the nucleus accumbens. And given that we now feel that we have an impaired circuit, perhaps driving it would actually make sense. So the question really is, can we restore inhibitory control and suppress compulsion in patients with severe obesity? So we applied for NIH funding and FDA approval, approval from CMS or Medicare, and applied to clinicaltrials.gov and designed this protocol where we would do just that. So we would place an electrode in the human nucleus accumbens with the goal that the stimulator would be able to anticipate by monitoring the nucleus accumbens electrical environment. And when this patient was craving perhaps that donut while he was supposed to be reading and enjoying his daily life, it would then stimulate to prevent the actual behavior from actually happening. And all of us can relate to this type of cartoon. Some of us might know some people that are actually dying of obesity because of this type of problem. So when I say dying, what I mean are patients that have at least morbid obesity, tried medication, behavioral therapy, talk therapy, and failed gastric bypass surgery, the most invasive option for patients with severe obesity. They had to exhibit a history of loss of control eating, which is pervasive in all binge eating related disorders, and also demonstrate compliance. We needed patients who were going to show up for their appointments, partake in food diaries, things of that nature. We obviously wanted to study loss of control eating in the most homogeneous way possible. So we excluded patients with severe, unstable psychiatric disorders and things of that nature. Our first patient was implanted right before the COVID pandemic started, by the way. This is somewhat representative. We've now implanted 3 patients. We've had the opportunity to study about 5 patients in a similar way, and I'll try to allude to some of those studies. One just got published in Neuron this week. But this patient had gastric bypass surgery about 20 years prior, had severe binge eating disorder, failed all treatments. And we implanted her end of 2020. The device that we used is a device that can be monitored using a HIPAA-compliant cloud-based database. So because of that, we were able to actually do a lot of our work remotely, which we had to do because of COVID. So at the very least, we were able to maintain some pace in our research. This was a milestone-driven grant, so we missed some of our milestones, but the NIH was fairly understanding of that because of the COVID pandemic. So lots of scientific findings have come from this. This is the first time we've been able to study the human nucleus accumbens in this way, using a device that's implanted and recording. But we needed to also make sure we were in the part of the nucleus accumbens that we felt was appetitive. And how would we confirm that? So when I first saw deep brain stimulation for Parkinson's disease by Dr. Baltuck at Pennsylvania Hospital, that strategy, which is fundamental in deep brain stimulation surgery, is to record. And we would monitor physiology of the subthalamic nucleus and listen for tremor cells. And you would literally watch the patient's tremor and hear an audible conversion of electrical signal that sounded a lot like what you heard. And it's just absolutely magical when you see it. I see it now 2 or 3 times a week, and it's still magical when I see it. And I see it every time we do it. We didn't know what the correlate to a tremor cell would sound like, look like, how to capture it, but we hypothesized that there would be appetitive units or craving cells that we could capture in the nucleus accumbens. And we designed an instrument, which is basically like a task, using personalized pictures that the patients would provide us that they found really appetizing. And as you probably know, when you're in the operating room as a patient, you are NPO, not able to have eaten any food, so you're very hungry. So it's a perfect state to study appetition. When we place the electrode in the brain, and this is sort of a visual representation of what we heard, when we entered the nucleus accumbens, we heard a lot of activity, but it exhibited no relationship to the pictures that we showed this patient. However, when we got to about 2 millimeters above the bottom of the nucleus accumbens, just 2 millimeters, when we showed a picture of, in this case it was split pea soup, we heard this activation signal, and we were able to capture that in a video that I can show you in a moment. this is our setup in the operating room. There's an intraoperative CAT scan machine. There's a patient in there. This is my team, a psychiatrist, an electrophysiologist, and Daniel, who is my imaging and neurostimulation expert. And you can see in blue, every time we showed a picture of a food item that the patient found appetizing, we heard activation. It was very consistent. We replicated this now in 2 out of the 3 patients that we've implanted in this clinical trial. And here's just one example. So we would show patients this sort of blurred image, kind of a neutral picture, not expect to hear anything. But then we would show this picture of homemade split pea soup. Our first patient actually did this, and she didn't like sweet foods. She liked salty foods. So Mexican food, split pea soup, homemade foods that she put a lot of bacon and lard in. And this is what we heard. It's all pretty quiet, a little bit of static. And then that picture goes away. We give her a moment of baseline, and then we show her the split pea soup. And the picture goes away, and then it's quiet again. And we were able to replicate this in a patient we just implanted actually at Penn about a month ago. So another goal of the surgery and theory that we could put to work if we needed to help these patients was that the device could stimulate the nucleus accumbens when needed and elevate the mood ever so briefly. We're actually pretty good at doing that. It's a lot harder to stimulate continuously and elevate the mood continuously, which is why a lot of deep brain stimulation trials for depression have failed. But when you do it intermittently, you sort of don't have that tolerance to the effect of the stimulation. The brain doesn't habituate to it. So we at least theorize that when patients binge or lose control, they have a moment of negative, what we call negative pre-meal affect. Right before they binge, they feel down or stressed or low energy. And so we wanted to be able to anticipate when that was happening and deliver stimulation to elevate that mood during that moment. And perhaps that would allow patients to sort of disconnect the moment of negative affect from the subsequent binge. And so I asked Nolan and Ian Walker, these are 2 psychiatrists that we work with at Stanford, great colleagues of mine, to join us in the operating room. And this is just a depiction, but what they were able to show is that a relatively low amount of current, 4 and here a rating of 6. This is 2 milliamps and here 1.5 milliamps. We were able to also induce an increase in mood in this patient. She started laughing in the operating room, for example. We don't necessarily require this result for every patient. We have 3 expectations in the surgical suite. One is that we can induce an appetitive response. The second is that we can induce a positive mood. The third, and perhaps most important, is that we've engaged this circuit of interest surgically. So we confirm that with that CAT scan machine in real time. And because this is a first-in-human study, and we're still exploring how to best do this, as a surgeon, I require 2 of the 3 of these things to be successful in order to say our surgery is now complete. Otherwise, you could be in the operating room for 12 hours. And we nearly are sometimes. So I'll keep moving here. The device that we used is a device that can record. It's completely implanted. There's nothing tethered or anything like that. And this patient is fully recovered. This is our first patient from the study. And what you can see here is a buffet of food. This was a buffet of food that was designed by Stuart Agris, who used to be at Penn a long time ago, to provoke binges. And one of his proteges, Deborah Safer, actually did a standard mood provocation in this patient to try to get her in the state where she would feel at risk for binging. There's no sound here, but you can see that after a standard breakfast and a standard lunch, by the way, wearing an eye tracker so we can see what her eyes are doing and exactly what she is looking at, and wearing a backpack here that allows us to actually track what is happening in the brain in real time and take timestamps in that trace so that we can do an offline analysis of what is happening in that brain at the moment of the bite. We're going back to the bite. And if I told you earlier,
I don't mean to be redundant, but this is a patient who had already eaten, and she's full. But this mood provocation was effective in inducing this binge. The FDA required that we allowed patients to binge for no more than 15 minutes, just for their own safety. That strategy is very similar to how we manage patients with epilepsy every day. So patients with epilepsy who have seizures, they get admitted to if they fail medication, of course, they get admitted to the hospital for monitoring, and we put an electrode on there, or an electrode array on their scalp. And we try to figure out where the seizures are coming from. We withdraw their seizure medications. We induce insomnia to a certain extent because they can't sleep well in the hospital. And then they have a seizure. And we video it, and then we do an offline analysis to figure out where the seizure was coming from and what we can do to stop it. Same strategy used here. Obviously, none of these patients enjoyed this experience, much like our epilepsy patients who have to have seizures, but they understand the relevance. So you can see here in the first patient, at the moment of the bite, thanks to that eye-tracking headset that we had, we could see that there's this increased power in that low-frequency range again. In mice, it stopped a little bit lower, like around 4 or 5 hertz. In humans, it went up a little bit higher to the theta range. Not sure exactly why, but obviously these are different species. But still a very powerful and parallel finding to what we saw in mice. And we saw it, by the way, in both of our first 2 patients. And again, very similar to what we had in mice some time ago. This device, remember, this was the COVID pandemic. So we were able to track her behavior from afar. And so we asked her to swipe with the device magnet that it came with right over the stimulator anytime she felt she was craving, anytime she felt hungry, and anytime she kind of just felt normal, baseline. And so she sort of would swipe pretty regularly. And over the course of about a year, we got an enormous amount of data. You can see here that, and these the red graphs here are representing moments of craving. And there's this clear dissociation between what the electrical signal looked like during those moments compared to when a patient felt hungry versus maybe just baseline. And it speaks to the fact that we can now, and by the way, we paired that with a food diary. That's why the patients had to demonstrate compliance, a very important feature of this study. We then programmed the device to detect this low-frequency signal automatically. And indeed, it would tell us it did these detections, but then we wanted to confirm that it actually detected what we asked it to, and indeed, it was very good at isolating these moments and stimulating in response to them. Our first patient, when we turned the device on for the first time, may have had a decrease in her frequency of loss of control eating in green, but it was mild at best, and she had no subjective benefit. By the way, this was all sham controlled as well. When we turned on the right side electrode, though, on the other side, we left the left turned on. She did have a relatively quick response. She texted me on her way home and told me a pretty incredible story, which we thought was very profound. And it was a fairly durable effect throughout the course of the study. And it was associated as well with a loss of body weight as well, as you can see. Not a tremendous loss in body weight, but remember, these are the most severe of patients, and we would expect their body weight to decrease slowly. Going back to her imaging, the right side electrode is engaged in that red structure. That is that ventromedial prefrontal cortex terminal within the nucleus accumbens. These are streamlines that we use in an indirect assay of those structures. But on the left-hand side, I intentionally missed it. So this is the structure we wanted to hit, and I'm over here on the right, about 2 millimeters lateral to it. And perhaps that explains why when we turned on the right side, she had this immediate effect. But when we turned on the left side, it was delayed. I missed it intentionally because you can't see it here, but in the caudate nucleus, just dorsal to it, there was a fairly prominent blood vessel that I could not go through. So we had to approximate our target. And in 3 dimensions, you can see how we missed the streamlines on the left side a little bit. We hypothesized that with a little bit of juice, we could capture them, but at a low dose, perhaps not. But on the right side, she had a wonderful immediate effect. The second patient we treated at Stanford when I was there had an immediate effect when we turned on the left side. And a little bit came back of the loss of control eating frequency in green, but when we activated the right side, the effect was reproduced. This was a more robust, a more rapid finding. And not surprisingly, both electrodes are sitting inside those streamlines. And it really speaks to, despite a very low sample size, the importance of this translational research and targeting this homologous circuit. We published this initial result in Nature Medicine. It was fairly profound that we were able to reach our primary endpoint within 6 months for both of these patients. This was a behavioral endpoint. We did not expect to have any changes in body weight in the short term, but we did. These were patients whose body weight returned to baseline before the device was turned on as well. We followed them to the end of the study. This was just a 6-month report. And this was not published in that article, but at 12 months, their body weight continued to lessen. Because of the interest of time, and I started late, I apologize. I'm going to skip the testimonials. I'd like to transition a little bit, quite literally, to my work at Penn, which I'll cover very briefly. It's all very much related. The palatial Stanford to the City School of Philadelphia, University of Pennsylvania. I lived at the Quad when I was an undergrad, and like I said, I spent a lot of time here in Study D. I was in Mask and Wig, and it was a performing arts group in college, and we rehearsed so much that I never had enough time to study. And the drummer was one of those fellows in Wharton. He was Ben Franklin's scholar, Joseph Wharton's scholar, that's what it was. Very smart guy. And he was the one that told me, he's like, come join me at Wharton tonight at Study D, and we'll study together. So that's why I spent a lot of time here. Plus, my girlfriend at the time also was in Wharton. So in any case, this is Penn. This is the room where I watched deep brain stimulation for the first time. This is me here. This is our first patient in the obesity study. This is our computer task here that she can see with these incredible little goggles. I think that's Wani Choi, who's right there. Hi, Wani. Wani is a bioengineering graduate student who plays the role of 3 postdocs in my research laboratory, quite literally. Wani, thank you for everything you do. This is my team, my clinical fellows on the other side of the street. That's Lee Min Q. I believe this is Nita, our research coordinator. Quite a team of people where we're trying to identify these appetitive units. A couple things I just want to point out. This is our first patient where we use tractography, that same technique, to identify in a personalized fashion where the sweet spot is in yellow. And these are the streamlines to the SGC, or the ventromedial prefrontal cortex. After surgery or in the operating room, I confirm accuracy, and in yellow are those same streamlines in a slightly different depiction, confirming that I was indeed an accurate surgeon that day. Of course, at the time of surgery, if I'm not accurate, I can always fix it. But after the fact, that's a little bit too late. What is really striking with our first patient at Penn is that when we implanted her, she was about 304 pounds. And yesterday, she was something like 276. So that is striking because that's more body weight loss than either of my patients at Stanford. She started a slightly higher body weight than the patients at Stanford, but really not much different. The other thing that's really striking is we have not turned her device on. So this is just an implant effect. In the world of neurosurgery, we discovered that stimulation can be helpful because when we used to place electrodes in the brain, they would disrupt tremor, seizures, chronic pain just by putting the electrode in that area. We call that a honeymoon effect. And we actually tell patients to look forward to the honeymoon effect, but recognize that it will be temporary. And it's probably a bit related to the implant. There is a little bit of microtrauma just putting the electrode in place. It probably does have a physiologic effect, though, because we've targeted the region of interest. Deep brain stimulation has been studied in double-blinded randomized controlled trials where patients get a sham surgery. They get an incision, they get a little burr hole in the skull, but no wire in the brain. but the patient would never know the difference. And in the patients where they don't get a wire, they don't have an implant effect. But in the patients that do get a wire, but perhaps the device is still off, they do get an implant effect. So we don't think that this is a placebo, but of course still low sample size. But still, whether it's placebo or not, my gosh, that is a wonderful result. Fortunately or unfortunately, because we're not able to activate her device, because we need her to actually be craving, and she's had such minimal craving since the surgery, I can't actually say, well, now we found the biomarker in the brain of craving, because she's not having it. So we are having a little bit of a slowdown with our NIH milestones, but as a sort of as an investigator who is also a doctor, the investigator in me is a little frustrated, kind of curious, but still frustrated. But the doctor in me is so happy for this patient because she's lost more body weight than she's had in a very long time. I'm going to transition and end as soon as I can to another disorder. This is OCD. I'm really interested in compulsion across diagnoses. And in a lot of ways, they're probably more similar than they are different. This compulsion is one of a dirty toothbrush. And we are telling this individual that this toothbrush was dropped on the floor, and we want him to imagine brushing his teeth with it. And you can see here, and I think you can hear it, this activation in cellular activity, similar to what I showed you with those appetitive units. Same brain region, same surgery, different patient, different problem, and a different type of provocation, or a different cue. Deep brain stimulation for OCD is actually the fundamental, foundational precedent that I used to convince the FDA to let me do deep brain stimulation for obesity. But deep brain stimulation for OCD is an available therapy. It is FDA approved under what we call an HDE, human device exemption, for rare conditions or rare needs. But at best, it takes the edge off. So a patient with severe OCD could get deep brain stimulation, but not necessarily see the benefit that they might want to see. Whereas a patient with severe Parkinson's disease, if they get deep brain stimulation, they don't feel like they have Parkinson's disease anymore. So I liken that to remission. They almost feel like they're in remission. Whereas in OCD, they don't remit. And at best, they are responders. So not to say we shouldn't be doing this, because I love to do it, we do it frequently, we need to do it better. Because we're already doing the surgery in OCD, though, because in obesity, this is brand new. But in OCD, we do this not all the time, but frequently enough. Surgeons like me do 5 to 10 of these a year, 1 a month, let's say. Since we already do it, we went to the FDA and said, hey, we think we can make it better. But we need to do a little bit more before we do less. So we proposed, and you can see here in all these different colors, monitoring the brain like we do in patients with epilepsy. So patients with epilepsy get what we call stereo EEG. At the Pavilion Hospital, in the epilepsy monitoring unit, there's probably about 5 patients there right now, with electrodes all over their brain that my colleague Isaac Chen places into areas that cause seizures. It's a very safe procedure. It's astonishingly safe. It is still brain surgery. However, we believe that we could leverage it to better understand circuits involved in OCD. It took about 3 years to put a protocol together that we all agreed to and decide on brain regions to actually target because they're somewhat innumerable. But indeed, we came to that agreement. And I use a standard deep brain stimulation contraption. I leverage the same phenomenal team that I have now here at Penn. I love these action shots. Thank you to my fellow for providing them. And we use a similar imaging strategy to try to target these areas. I won't go into too much depth, but suffice it to say that we actually implant 16 electrodes in the brain into most of the areas that we believe have been rigorously studied in either animal studies or human imaging studies, as well as deep brain stimulation studies in humans, to suggest promise in managing patients with OCD. But we want to do it better, and we believe it has to be personalized. This is our first patient. This trial got FDA approved. It's funded by a foundation started by donors who are parents of children with OCD. One of them I took care of when I was at Stanford. And you can see here And this patient obviously consented to this. And you can see at the very, over here, you can see the room setup and the electrodes here. These are all externalized, unlike that other device I showed you earlier, because this is a temporary procedure that we do, just like patients with epilepsy. And this is all done at Pennsylvania Hospital, which I'm very proud of. On the top here is us assessing this patient with the device shut off, or with the electrode shut off. No stimulation is being delivered. This is sham. And you can sort of just see his fairly flat affect. If you weren't blinking, you wouldn't even know that this was working. So this is his kind of baseline affect.
Okay.
And with
The
stimulation,
Shire. Up and down. With butterbeers from Harry Potter.
Oh, wow. Nice.
Big ham coming by for you. I see you drinking nice on the table. Pork bellies, everywhere. A pork roasting on the steak.
so he's joking around with my team. But, this happened very recently, so we're not deep into the analysis of everything. I should say we are deep into the analysis, but I'm not prepared to report it all to you. But suffice it to say that of the 16 electrodes that we placed, 6 of them exhibited this type of therapeutic promise. But only 4 of them seemed to hold up to sham testing and demonstrate reproducibility. And so he has subsequently actually had another surgery. We took out these electrodes and then placed a permanent deep brain stimulation system targeted to those areas alone. And one reason I was late today was that I was programming his device with my psychiatry team. We do it all together. So I mentioned that when I started that I would circle back to Pennsylvania Hospital. Pennsylvania Hospital, well, everyone knows about this amazing pavilion. Pennsylvania Hospital, I would argue, is the historic powerhouse of Philadelphia, founded by Ben Franklin and Thomas Bond. But actually, I would encourage all of you to do a little bit of research on this individual. Does anybody know who this is? This is Benjamin Rush, the only doctor who signed the Declaration of Independence. He was John James's best friend, and he created this building here, the West Wing, so they called it at the time, which looks a lot like Pennsylvania Hospital. This is Pennsylvania Hospital. He wasn't the founder of it, but he was the brilliant doctor that saw the need to have this separate facility that then became the main hospital for what they called the insane. These were patients who lived at Pennsylvania Hospital in the basement, technically SB, where the morgue is now. And they lived there for years. One of those patients was his son, Jonathan Rush. This was all in the 1800s, late 1700s, early 1800s. And he was the only doctor in America at the time, and then inspired doctors in Europe and abroad, Canada, to acknowledge that these were not patients that were insane. They were patients that were sick. And he actually developed therapies. I can't say we use them today, but therapies at the time to try to get these people not just upstairs, but actually out of the hospital. There's a biography that is relatively recent by Stephen Fried that I just finished that I think it was 1,200 pages. And I'm not one of those voracious readers that has to read all these types of things. But my gosh, we love Hamilton, the Broadway show. I don't know if anybody here has seen Hamilton. The sort of forgotten forefather. He's not the forgotten forefather. This guy is. There's so many things that he did that he gets no credit for. This is definitely one of them. The other thing is that he actually articulated common sense to Thomas Paine. And Thomas Paine wrote it. And the reason Thomas Paine did that was because Benjamin Rush was a young doctor at the time, and he didn't want all of the wealthy politicians in Philadelphia to think that he was an abolitionist. So that's why his name is not on the common sense authorship list. In any case, I promised myself I would always end with a little segment about Benjamin Rush when I talk about psychiatric research at Pennsylvania Hospital. If we have a little bit of time, I'd be glad to take questions. I hope I didn't overstay my welcome.
That was fantastic. Yeah. Okay.